Hello everyone, welcome to Get My Class. The question says which of the following is a selectable marker? The first one they have given beta galactosidase, and the second one they have given ampicillin resistant G. And the options they have given both one and two. Option B only one. Option C only two. Option D none of those. So let us understand the question. It says which of the following is a selectable marker? Okay, so what do you understand by selectable marker? Selectable marker. These are the markers which help in identifying the transformants. Okay, identifying the transformants. So what do you mean by transformants? The transformants are the ones which contain the rDNA, that is recombinant DNA, or you can say the gene of interest. And where is that gene of interest? Within the vector. So within the vector. Now this vector should have selectable markers, okay, which will help us to identify the transformants, because this vector will also carry the foreign DNA, right? The gene of interest. Okay. Now here they have given the option as beta galactosidase. So beta galactosidase, what is that? Beta galactosidase, it is gene. Okay, beta galactosidase is actually an enzyme, if you see, which will help in breaking down the lactose. Okay, it helps in breaking down lactose, and if it is present, it will give rise to blue colonies. Okay. The organism which is having the vector containing beta galactosidase will show, it will be blue colonies. Okay, so this will, of course, those vectors will contain this gene which will code for that. So hence this can be considered as a selectable marker. The second one says ampicillin resistance gene. Ampicillin, or you can say AMP-R gene, I would say. Ampicillin resistant gene. Now ampicillin resistant gene, it clearly indicates that it is resistant to ampicillin. The organism here, if it has this gene, it will be resistant to ampicillin. So what will happen? Then it will definitely grow in the medium in the presence of ampicillin. Okay, it will grow in the presence of ampicillin. Why? Because it has resistant gene, so it will not get killed. Whereas the non-transformants will not grow in the presence of ampicillin. So that's how this also can be considered as selectable marker. So option A says both 1 and 2. That is the perfect answer. Whereas option B says only 1. It is ruled out. Only 2 is ruled out. None of this again ruled out, because we have both, that is beta galactosidase and ampicillin resistant gene. Both are selectable markers. Okay. So let us conclude the answer. The answer is option A. I hope you understood the explanation. Thank you, everyone.